variety of projects. Of course, our work focused on osteoarthritis, but NASA and NIH were particularly interested in diseases and pathologies that affect astronauts up in space, but at the same time, research that would benefit people on Earth. So the work, of course, is applicable to people everywhere, whether they're on Earth or in space.
Yeah.
And it turned out that, as we had known for a while, the research that we got involved with could most successfully be approached by working with real human tissue. And it turns out that the project, of course, astronauts, as I'm sure you could imagine, are the most highly physically trained specimens on the planet.
Mm-hmm.
They're extraordinarily in good shape, even though quite a few of them have had joint problems in one way or another, but they're all very highly trained physically. Nevertheless, it turns out that injuries during the mission period up in the space station occur more often than injuries, orthopedic injuries, to us on Earth. And so the NASA and the astronauts are very interested in understanding these problems. So we've begun to work on this, and the rationale for human tissue is that while there are some animal models, they're just not good replicates of the real human disease.
Do it without studying the patient. So I think it's really critical. And as I mentioned, one of the reason I chose breast cancer is because we have access to normal, early stage, later stage tumors that are really important because we want to study the patient. We use experimental models also, but I feel like, for the discovery part of the projects, we want to do something that we know is in the patient, because that means that it's real. And, I always say, like, the experiment has been done. We just have to figure out what was there. And then also, we would like to, if we're doing experiment and experimental models, you never know what really happens. We know what could happen,
Mm-hmm.
but if you're studying the human tissues, then you see that that's what's happened there. So I think it's kind of, different people have different approach to how they do research, but to me, like, studying the patient is really important, and we cannot do it without the human tissues. And pretty much all of our projects is using human tissues at different phases. Sometimes we use it for the discovery part. Sometimes we use it for validation, like, for example, in the papers we recently published, when we're doing like a CRISPR screen to discover new targets, but then we go back to the clinical samples to validate those. In other cases, we do profiling of the clinical sample, patient sample, and then we do experiment in an experimental model to validate hypotheses that we build based on the tissue studies. So it's, I don't think we have any project that would not use human tissues in one way or the other.
Our work is almost exclusively done in human tissue in the development of human cell models. And our rationale for that is essentially we take out the complexities of interspecies responses. Is a mouse really, is that comparable to the human disease state? By using human cells in human tissue, we obviate the need to demonstrate relevance. Mirroring models are important, don't get me wrong, and we do mirroring models, but they often fall short in the characterization of the nuances of human disease. It's really requiring human tissue and human cells to be able to do that. And we've been successful in doing that for decades, really.
We grow the cells from human lungs in order to study lung diseases. And in 2001, I became the director of this core facility that provided both cystic fibrosis and non-cystic fibrosis lung cells to researchers. And it's been a very interesting journey. And it's been really quite remarkable because as a result of the work and the provision of these cells, we've really made very significant advances in the treatment of cystic fibrosis. And I feel a little bit of career satisfaction in that we helped provide the materials that were the basis on which these drugs, small molecule drugs that treat the underlying cause, not just the symptoms of cystic fibrosis. And it's been really an amazing experience. And that extends not only to cystic fibrosis, which is a rare lung disease, but to other lung diseases that are much more common, for example, asthma or chronic obstructive pulmonary disease. And we contribute cells to many different studies that address key issues in these very important lung diseases that are cause of a lot of pain and suffering. And in the last 10 years, we've actually been studying coronaviruses with these same cells. And we were right on the spot when the SARS-CoV-2 COVID-19 pandemic hit. And we've been able to contribute these cells to many studies about the basic biology of that lung infection, and also to help understand some of the treatments. And that also has been very satisfying in terms of career accomplishments. And NDRI has been a partner through all of these years, helping organize and provide and take advantage of the lungs that are available that don't quite make the cut for transplant for some reason, an inappropriate donor match or other issues that prevent them from being used for transplant. And in that case, if the families sign off on the ability to use those lungs for research, they can be provided to researchers. And, you know, that's also been a source of satisfaction because organs that people generously donate with the hope that they'd be transplanted, but they're not really appropriate for transplantation, they could be used for research. And that research can, just as the family's wish was that the organs of their loved ones could help other people, they are helping other people, but in a different way. So NDRI has helped us on this journey, and we very much appreciate their service and the many years of working with them.